the Genominon webinar, Automated Biomarker Selection for Evidence-Based Gene Panel Design. I'm Candace Chapman, Director of Marketing for Genominon, and I'll be your host today as we walk through a better and faster method for identifying biomarkers for use in panels by utilizing an evidence-based target identification process. This method uses the Mastermind Genomic Search Engine to objectively correlate genes and genetic variants with the quality and frequency of citations from the scientific literature. Dr. Mark Keel will demonstrate the process with a real-life example of a gene panel he created for hematopoietic malignancies. As you are watching, please feel free to submit questions to Dr. Keel through the chat window on the right or on Twitter at Genominon. So without further ado, I'll introduce our speaker. Dr. Keel is founder and chief science officer here at Genominon, where he oversees the company's scientific direction and product development. Prior to starting Genominon, Mark completed his residency in clinical pathology in 2014 at the University of Michigan. While at Michigan, he completed a fellowship in molecular diagnostics and devised the informatics framework for clinical next-generation sequencing in the molecular diagnostics laboratory. During his doctoral studies, he made groundbreaking contributions to the study of hematopoietic stem cells, for which he was awarded the Weintraub International Graduate Student Award and the ProQuest Distinguished Dissertation Award. While a postdoctoral researcher, he made significant contributions to the field of hematopathology, including genomic profiling of lymphoid malignancies, for which he was awarded the Benjamin Castleman Award. So Mark, I will let you take it away.
Thank you, Candace, and thank you, attendees, and welcome. I'm Mark Keel, a co-founder of Genominon and the Chief Science Officer. The problem that we intend to discuss today is one of biomarker selection for next-generation sequencing gene panel design. This is the content selection aspect of panel design, one of the most challenging. It's challenging because it is extremely time-consuming and can take months of labor-intensive searches to find the appropriate empirical data for biomarker selection. And this can take months because of information overload. The source of truth, the source of that information is in the primary medical literature. There are over 3 million titles and abstracts in PubMed, and over 6 million of those contain information relevant to genetics and genomics. And searching through those and prioritizing the most meaningful can be extremely overwhelming. Moreover, when making informed content selection decisions for designing next generation sequencing panels, oftentimes expertise is required that may not exist on teams charged with designing those panels. And so in some, decision makers, those who have to finally adjudicate the candidates for gene panel design, often lack objective evidence for content selection. These current approaches of manually reviewing and searching through the literature to identify candidate biomarkers for gene panels are inadequate, as consequential biomarkers can often be missed in the rush to summarize information from the literature, as opposed to performing an exhaustive scan of the literature to identify any and all relevant biomarkers, as well as frequently inconsequential biomarkers taking up space on your gene panels, being as they are not backed by clinical evidence. And even those biomarkers that are consequential, it's often a significant challenge to identify the most relevant reference citations to justify those conclusions. So we at Genominon, in working with several of our software clients, have heard of the need for solving these challenges in content selection for gene panel design, requiring, as they do, a more evidence-based approach to biomarker selection. So for those of you who aren't familiar with Genominon or Mastermind, Mastermind is the backbone of Genominon's solution set. It is a comprehensive genomic search engine software that's predicated on the genetic literature. And the way that we have built Mastermind is by indexing the genomic literature, including those 3 million titles and abstracts from PubMed, from which we have prioritized the most meaningful full text content for further indexing to identify the mention of any one of thousands of different diseases, any one of tens of thousands of different genes, or any one of their synonyms. And once a gene is identified in any paper of merit, indexing the rest of that full text article to identify any variant in any transcript of any one of those genes, no matter how they may be described, using conventional HGVS nomenclature or not, at the cDNA or protein or RSID level. So the content composition of the Mastermind Genomic Database is over 6 million full text, fully indexed genetic and genomic articles. This database is updated on a weekly basis, and to date we have over 1.5 million biomarker candidates and all of their relevant disease associations. So what I want to spend the webinar time talking about today is our process of evidence-based biomarker selection, leveraging the data content and database querying infrastructure internal to Genomenon for accessing this information within Mastermind. So the way that we proceed in engaging with clients who are interested in content selection for next generation sequencing panels is we begin with a formulated query of the Mastermind genetic database. That query, as I'll discuss later, can be a disease or a set of diseases, or otherwise a predetermined set of genes. The evidence for inclusion on a panel may be lacking, and that may be what the query of Mastermind is intended to produce. So from that initial input, after a conversation with our clients, we automatically produce a comprehensive biomarker list of genes and or gene variants for any one of those diseases or disease groups of interest for that particular purpose. Those biomarker candidates can then be filtered, prioritized, and refined according to specific clinical or research goals. Examples of those include, is this biomarker relevant for diagnostic purposes in clinical workflows? Is there prognostic significance to that biomarker in the context of a given disease? Or are there relevant therapeutic concerns associated with that biomarker, including whether there is a specific drug or compound that has been linked to the biomarker and possibly a disease of interest in any one of the millions of references comprising the Mastermind database. So once that database, that sort of core database has been assembled, all of the evidence that is extracted out of the Mastermind database can be ranked by number of citations and the strength of those associations, including at the sentence level, identifying in the full text where biomarkers, diseases, and clinically significant key terms may be co-mentioned in a sentence, a paragraph, or a section of any one of these millions of articles. In combination with all of that information at the granular level, as well as high-level information such as the journal, journal type, the date of publication, as well as the number of citations that that individual article has received, that information is then assembled to prioritize both the biomarker candidates in the process of assembling this core database, as well as prioritizing individual reference citations that specifically justify the case for that biomarker being included on a client panel. And then finally, the end process or end procedure for assembling this database is an internal manual review and quality assurance process. So there are two distinct use cases. We've had about an equal measure of call for type 1 and type 2 from our current customers. One is Denovo target selection, and the other is biomarker validation. So de novo target selection is where a client comes to Genominon with a set of target diseases and all biomarker candidates, and they wish to prioritize those biomarker candidates according to the evidence predicate in Mastermind that I described on the previous slide. The other scenario is where clients come to us with a panel that they have already designed, and they wish to justify the conclusions of that panel and inclusion of separate biomarkers on that panel by extracting information and organizing evidence out of Mastermind. So situation one is useful for groups who may lack experience or may not have much working knowledge of different diseases or the genes associated with these diseases, or otherwise may be newly promulgating a panel in their diagnostic workflow. And Mastermind can help with a simple input of a disease or set of diseases and can produce the list of genes as well as all of the variant biomarker candidates with all the reference citations prioritized according to the customer's taste. And number two is where the panel has already existed and customers are interested in providing support for marketing initiatives or otherwise for providing evidence to payers to justify the inclusion of those biomarkers on their panel. So along either of those two lines, there are several example projects that Genominon has recently worked on that I'd like to exemplify here. On the left, one of our earliest panel design projects was initiated by one of our software clients who was previously searching iteratively through the Mastermind software for biomarker candidates to be included on an acute myeloid leukemia companion diagnostic panel. And in conversation with them at some point during their license to the software, we realized that we could help facilitate a faster production of this candidate biomarker list to inform their acute myeloid leukemia panel. And again, that was all the way down to at the variant level for each of hundreds of different genes that have been linked to acute myeloid leukemia in the literature. So that was one of the first panels that we had designed for our clients. And more recently, in the middle three here, are a unique breadth of different panels that we have designed. The first that I'll describe here is DNA damage repair and genes that are associated with that phenomena and are relevant to clinical concerns for a diagnostic panel to be used for third-party reference laboratories. So that was a scenario where we began with a list of genes and an understanding that the focus of the panel was on this DNA damage repair disease set, and Mastermind was utilized to prioritize from among that existing panel of genes which genes should not be included. There was not sufficient evidence to justify inclusion of those genes on the panel, and which genes most certainly should be included based on evidence support and strength of their association with this disease type from the medical literature. Another recent panel design project that we've worked on similarly involves extracting information at the variant level, this time not for malignancies, but rather for genetic obesity screening. So this project began different from the DNA damage repair project, which had a list of genes predetermined. This panel required us at Genominon interrogating Mastermind to come up with a list of genes de novo from a set of 6 core obesity-related diseases. And again, this is in contradistinction to the DNA damage repair, which was predominantly a somatic panel. This genetic obesity screening panel was for hereditary diagnostics. And then also, on the other end of the spectrum is a direct-to-consumer array, where Genominon is using Mastermind to identify SNPs or RSIDs or variant biomarkers that have been linked to enhanced athletic performance and other exercise physiology related phenomena such as diabetes, VO2 max, and obesity. And so this is an array, it's a more focused array predicated on individual polymorphisms and identifying the core evidence to support inclusion of hundreds of different SNPs on this direct-to-consumer array, and providing all of the necessary evidence to support conclusion or justification of those biomarkers being included on that panel. The last panel on the right is a panel that we had designed internally at the request of a number of our clients. We decided to design a Pan-Hematopoietic Malignancy Panel, the process and outcome of which I'll be walking through for the last part of this webinar. This began with a desire to extract from Mastermind relevant biomarkers informing disease pathogenesis for leukemia and lymphoma broadly. So rather than being a very focused panel, this was a much broader panel where we were casting a wide net and assembling all of the evidence necessary to produce a prioritized list of biomarkers for hematopoietic malignancy. So in total, using Mastermind, we were able to identify more than 500 genes that were prioritized from the literature based on the number of citations and the cogency of those associations in the literature. We provided relevant clinical annotations that I'll walk through here in a moment by showcasing the deliverable for this final panel design, as well as providing justification in the form of prioritized articles and references cited, so that the final deliverable was all of the information needed for decision makers selecting the content to comprise genetic panels. All of that information was provided in the final deliverable in a readily searchable and prioritized way. And all of these biomarker candidates, as well as those prioritized references, had hyperlinks in the final deliverable back into the Mastermind software itself, so that users could see the work that Genominon undertook to produce this final biomarker list. And so the process that we typically invoke, and that in particular we invoked for this Pan-Hematopoietic Malignancy panel, is described in this Genominon white paper, which you can access by going to the website that I'm showcasing here from our homepage. It's White Papers. And you can actually see the biomarker candidates for both leukemia and lymphoma. And if you're interested, you can see the final deliverable if you inquire to support@genominon.com. We'll provide you with access to all of the information that I'm about to show you on these subsequent slides. So this is an example of the types of deliverables that are provided during Genominon's panel design service work. And I'll try to walk through this slowly. I apologize that the text is fairly small. But over on the left, at the gene level, you can see every gene candidate in a prioritized order. All of those candidate genes for biomarker inclusion, I think this is for the leukemia panel. And then just to the right of those genes are links into Mastermind, and those links I'll showcase the software aspect that those links go out to in Mastermind. The first is to showcase what specific hematopoietic diseases or leukemias each of these individual biomarkers have been associated with, such as T prolymphocytic leukemia or acute myeloid leukemia. And then the link on the right will actually take you into an overview of the way that that gene has been associated with leukemia or lymphoma in the literature by showcasing some of the data content in Mastermind, as I'll show in some screenshots here momentarily. The next section of this deliverable, where you see X's, characterizes the nature of the biomarker relationship to a specific disease in the way that this gene may be mutated, the gene may be over or underexpressed, leading to the pathogenic mechanism of disease association. There may be copy number gains or copy number losses associated with development of disease at the hand of that biomarker, or otherwise there may be recurrent translocations or structural alterations that lead to the association of that biomarker in the development of this specific disease, or a combination of any of those things. And depending on how our clients intend to use their final diagnostic or research-based panel, we can return or relatively prioritize different types of genetic associations, be they mutations or translocation events or other. The heat map there in the middle of this deliverable is a reflection of the content of the evidence and the strength of that evidence. The dark blue on the right is in summary form, where we have a non-parametric integrated score of all of the information and evidence from the Mastermind database, again, after having had a conversation with the client to understand what their needs are. Some of those lines of summary include whether the gene is mentioned in the title, and if so, in how many references, and whether the disease too is mentioned in the title or the abstract, or otherwise whether the biomarker, the disease, or the specific variant are mentioned in the full text, how often they're mentioned, and whether they're co-mentioned at the sentence level or paragraph or section level in concert with clinically relevant terms that are germane to the concerns of the client for the final panel. So examples of those would include prognostic significance, such as Kaplan-Meier survival data, therapeutic decision-making, such as generic therapy-related terms or otherwise specific drugs mentioned adjacent to any of these biomarkers, or otherwise general diagnostic utility of the biomarker. And all of that information gets integrated and prioritized, and what you're seeing here is the tip of the iceberg of all of the data that we had accumulated. In total, many thousands of biomarkers were interrogated, and in the end, a couple hundred were included in the final panel design. And then I think lastly, and perhaps most importantly, is a set of highly prioritized reference citations to justify that biomarker's association with this particular disease. So you'll see a series of numbers that are blue. Those are actually hyperlinks into the specific references that are listed to the right of those blue columns by title. The numbers there are the PubMed identification numbers or article reference number. And they're blue because they actually hyperlink into the Mastermind software itself for that biomarker in association with the disease of interest to get you directly to that reference so that you have unfettered access to all of the evidence that the Genominon team and this Mastermind panel design process has assembled for you automatically. And typically, from start to finish, this process will take our team between 2 and 4 weeks, depending on the complexity of the prioritization and annotation, as well as the sheer computational needs predicated on how broad-reaching the disease sets are, or otherwise how long and well-studied the list of genes may be. But in the end, what you're seeing here is a distilled summary of the candidate biomarkers and all of the evidence that's necessary for making informed decisions about final gene panel composition conclusions. So just to get you a window into the software for those of you who aren't familiar, this is the result of one of those links that I described in the deliverable of the panel design. This is a search on one of those biomarkers. It's a methyltransferase DNMT3A, which is well known to be associated with leukemia and has prognostic and therapeutic significance. If you search on that gene or otherwise follow the hyperlink provided in the final panel deliverable, what you see on the left are all of the specific types of leukemias or otherwise corresponding or associated diseases or biological phenomena where DNMT3A has been associated in the literature. And then in the middle, you can see an itemization of the number of articles that co-mention any of the diseases on the left and the gene on the right. And though that data, that evidence, that information is provided comprehensively for every gene and for every disease, as well as for every variant in any one of those genes throughout Mastermind. This is a reflection of the data content of Mastermind. On the right is a summary of the article landscape supporting the conclusion that DNMT3A is associated with the disease leukemia. The article plot, at the upper right, depicts at a graphical level the literature landscape of these citations. On the x-axis, you can see the publication date of each of these individual articles, symbolized here by circles. The size of those circles is the relevance and cogency of the evidence that the gene and disease, or otherwise variant and disease, are co-associated. And on the y-axis is the citation score or impact factor of any one of those individual journals. Below that, that information is provided through the software in list form. And then on the left is a summary of the variant landscape for each of these genes on this terminal page of Mastermind, where you can see along the linear axis of the protein how the mutations have piled up in different functional domains or hotspots of this protein as they're mutated and associated with a particular disease. I'll point out that the height of those bars for each of the variants is on a log scale, so you can very quickly see what variants are extremely well characterized, but also get a window into which variants may have only been mentioned once, and in the full text of any one of these millions of references that justify this disease and gene association. And any one of those variants can be selected for filtration and reprioritization of the article content and evidence information provided in the Mastermind software. Then this is another link that you would follow from the example Pan-Hematopoietic panel design that I showcased earlier. This is actually showcasing within the Mastermind interface that you can open up and visualize the full text PDF of these prioritized articles. On the left, you can see the highlighted abstract for an individual citation. And then I'm not showing this, but critically important is that all of the sentence fragments that mention the gene, the variant, or any clinically relevant key terms from the full text of any one of these references, that information is provided for all users of Mastermind, whether they have access to the PDF or not, so that very quickly they can scroll through that information for an individual article, or otherwise across multiple articles, they can rapidly toggle through the prioritized evidence and get a window into why that variant or that gene is important for this disease or that clinical circumstance within a given clinical indication. So to summarize what I showcased here, the benefits of invoking Genominon's gene panel design service utilizing the Mastermind database is that this process proceeds very quickly compared to some of our clients have been complaining that prior to engaging Genominon and this Mastermind panel design process, their content selection process required many man-months of effort with an unsatisfactory conclusion at the end, being as it was that they didn't have a comprehensive compendium of evidence from the literature to support their ultimate conclusions. In contrast, Mastermind can produce these outputs in a matter of days. As I said, the turnaround time is typically less than 4 weeks, even if the client has no previous knowledge or working experience with the disease or diseases of interest. Just as importantly, perhaps more importantly than the speed with which we can produce this data is the accuracy and validity of that data. So the results are more comprehensive as we have a very broad and deep reach into the medical literature, comprehensive coverage of the full text of many millions of scientific articles not covered by any other tools, as well as a comprehensive reach into the whole spectrum of human disease and the entirety of the human genome and every variant that exists in every one of the transcripts of every one of those genes. If those variants are published in any one of the 6 million articles in our literature canon, that information is prioritized and provided to you in a ranked order. And then finally, and I think critically, all of the information that is provided to our clients is justifiable and backed up by the supporting literature citations. So in essence, clients can see the work and the pains that were undertaken to develop this final panel design, either at the gene level or the biomarker, in the individual variant level. References that are prioritized according to those content specifications are all provided to you in the final deliverable. Just a little bit before I pass it over to Candace to field any questions that we may have received, a little bit about Genominon. As I suggested, I'm one of the co-founders of Genominon. That co-founding occurred in 2014. I'm originally a molecular diagnostician by training from the University of Michigan. We're currently headquartered in Ann Arbor, Michigan. The Mastermind product, after many years of incubation and refinement, was launched in the spring of 2017. And in that short time, we've already accumulated a user base of over 1,000 variant scientists, geneticists, and pathologists in more than 30 countries. We have a number of clients who are utilizing our panel design services, as well as taking advantage of some of our API capabilities. And we also are very supportive of some of these large-scale variant curation initiatives such as ClinGen. So with that, I think I'll pass it over to Candace. I'll thank you for your attention, and I'll be happy to field any questions that you may have.
Thank you, Mark. Now let's get some of these questions answered. But before that, I just wanted to know, if you have not already requested a Mastermind account, you can do so at mastermind.genominon.com. It's free. And hold on a second. You need to use the invitation code Pan-Hem when it's asked for. And this will give you a license for the Mastermind Free Edition. And we'll also start you out with 30 days of Mastermind Professional Edition to try. So you have answered several of the questions that came through, which is awesome. There was several questions about the time, how long it takes to get a gene panel design product, and you know, versus other methods, and how much money does it save, or how much time does it save. I think you've answered that, but if you have anything else to add to that, we can talk about that.
Yeah, so I'll just go back and emphasize a couple of points. So in the process, one of the most important aspects of the panel design process is the initial engagement with the client. To really understand what the final panel will be used for, and what the purpose of the panel is, and how that data should be best prioritized. And so we've done this enough times before that we see patterns, and we understand how to elicit the needed information from that conversation. And very quickly are we able to take the customized aspects of this panel design approach and put that into our automated process to produce an initial curatable database of biomarkers that then goes through the rest of our process and final QA and QC. And so all told, we can turn around these panels, even if they're fairly large, within a matter of weeks, some as short as 2 weeks even.
Okay, great. Oh, and we have a question here. When you were on the panel deliverable slide, Daniel asked, what do you mean by consequential biomarker?
So that's a great question, and it allows me to follow up on what I just described. So consequential is in the eye of the panel designer, and so that's where we really, I think the whole process benefits greatly from that initial conversation. So when I say consequential, it depends on what the client is intending to use that panel for. For instance, if the panel is intended simply for marrying the biomarker with specific therapeutics. If there is a biomarker, a variant, that is recurrently associated with a disease, but for which there is no targeted therapy, that is not consequential in that context. In contrast, if some other group is interested in designing a panel around the exact same disease, but is not as focused on or interested in therapy, but rather in making diagnostic calls based on the presence of that variant in a patient sample, well, that variant that otherwise didn't have a therapy associated with it, and the references that pointed to that variant, that would be a consequential biomarker and would merit inclusion on that client's final panel. And so just again, to take you behind the scenes a bit into our QA/QC process, the initial database assembly is a highly automated process, and we have automated aspects to the final QA/QC process. But if we're invoking variant level panel design, each of these variants is individually interrogated by examining in several instances every sentence from any one of these references in which they appear, and all of the annotated clinically relevant key terms that may co-appear in any one of those sentences. And so again, it's a hybrid sort of automated and manual process of QA/QC, and we really, as I say, we really have the process benefit from that initial conversation, so we know along what lines to prioritize these variants and these references, and how precisely to define what consequential means.
Awesome. This question from Sharon is, I think, related. Have you had incidents when the software prioritized a gene or a variant, and when manually checking it, it was found to be less significant?
Yes, absolutely. And so that's the reason that we invoke the final manual review process. We have a highly technical process of identifying this content from the literature. And for those of you who aren't familiar with text and data mining, there are a number of attendant technical challenges and limitations. There's attempts to solve those technical limitations with something called natural language processing. And Genominon and Mastermind has taken a different track. Those challenges associated with understanding the native language describing the variant and its associations with specific diseases, those are suboptimal, to the point that making any determinations using a solely automated approach is often riddled with error. And so rather than having the automation do all of that work for us, we have the automation do the heavy lifting, and then we organize and mobilize the evidence necessary to do the nuanced interpretation and finessing of the information in the form of manual review. And that manual review pulls out a number of patterns for false positive matches, and we leverage our understanding of those patterns to enhance the efficacy and efficiency of that manual review. But certainly the automated initial database produces false positives that are then weeded out in the final QA/QC.
Great, thank you. So James is asking, how can Mastermind be used to keep panels up to date? Can you use it to analyze the results of the panel? That might be two questions.
So great question. I'll answer the latter one first. So almost certainly, if you have a gene panel that was designed either using Mastermind's approach or not, in clinical practice, any of the variants that are seen in that panel can be adjudicated using the Mastermind software by searching for an individual variant to identify the reference or references and the clinical context in which that variant appears in the literature. So to be sure, any of the panels that we have designed the users of those panels who have access to Mastermind will be using that software to perform interpretations of those variants. The first question was about keeping the panel up to date. We've heard this referred to as panel landscaping, either understanding what new information was recently published or is recently now coming to the attention of practicing clinicians and therefore warrants a position on a diagnostic panel, or otherwise to go through pre-existing panels. In some of the cases for clients that we've worked with, this amounts to dozens or even hundreds of panels, and invoke a semi-automated or a highly automated process of identifying which of those panels contain biomarkers for which there's very little evidence support in the literature, where those panels can then be manicured to get deeper coverage in your sequencing results by virtue of having fewer biomarkers on your final panel, or otherwise eliminating the risk that you identify passenger mutations from a bloated panel that includes biomarkers that have no relevance to the disease, at least not consequential relevance, and that instead in the patient's results will just bog down interpretation and curation activities. And so that aspect of keeping your panel up to date or otherwise pruning out irrelevant candidates that had predated engagement with Genominon and the Mastermind technique, we've done that for a number of clients in the past.
Great, thank you. Here's another question in the chat. Can you design panels around non-clinical terms of interest, e.g. terms that are not medical subject heading terms?
That's a great question, and perhaps I didn't spend enough time in some of those example cases. I'm particularly referring to the direct-to-consumer athletic panel that we have recently designed. So for that process, there were a number of terms and conditions that weren't properly diseases, which comprise the backbone of the Mastermind gene ontology. And so we added those new terms into Mastermind itself, and then reprocessed or re-indexed our reference material to newly identify genes that may be associated with those conditions that weren't previously part of the Mastermind database. And so those can be free text. We can be provided with that information up front. We like to have conversations with the clients about what it is exactly that they're looking for, understand what synonyms or what are the best terms to be invoked to best identify the terms and conditions of interest. Once we've settled on and established a dictionary of terms and their synonyms, all 6 million of our references are re-indexed, and new disease or condition gene associations will emerge out of that process. And so that's a fairly straightforward operation that we invoke for clients who are interested in not properly clinical or medical associations.
Very good. So I am not going to pronounce this correctly, but Xiaochun Bai says, using the invitation code, do I have the access to the professional version for looking for multiple genes related to one disease?
So that's a great question, and we're on the right slide to answer it. So the professional version, which you will have access to for the first month of your use of Mastermind, allows for multi-parametric searches, searching either by multiple diseases, multiple genes, multiple variants, or otherwise performing free text searches. The diseases, genes, and variants can be united through AND operators or otherwise using OR operators to have a very specific or a more broad search. That feature is available in the professional version, which you will have access to for the first 30 days of your engagement.
Okay, and for your panel design projects, do you take into account other lines of evidence, such as population, frequency, algorithmic predictors of pathogenicity?
A good question. So very recently, we have had a number of clients interested in a refined QA/QC process at the end of the initial biomarker panel assembly. And that involves assessing the pathogenicity of each of these variants according to either ACMG variant classification guidelines or otherwise the AMP variant classification criteria. And so, as you may know, the ACMG classification criteria requires a couple of lines of evidence to interpret the variants. One of those lines is population frequency. Another is in silico predictors or computational predictors of damage or pathogenicity at the protein level. And then obviously the third, and what Mastermind really specializes in, is any additional evidence, often very consequential evidence from the literature along functional consequence or segregation patterns or otherwise identifying case reports. And so for those clients who are interested in ACMG and AMP classification of their variants in the database, we do pull in population frequency. We do invoke some of the data from these in silico prediction databases, and then we harmonize that information and holistically interpret those variants using the evidence from Mastermind's literature content. And so the process of doing that is obviously more involved than assembling the initial biomarker database and performing routine QA/QC, but it's definitely something that we have in our capability.
Okay, so Elena asked, does Mastermind include the supplemental material that can be included with PubMed? And what about geodataset information?
So that's a good question. It would be great if I could answer in the affirmative. So we at Genominon have for several months now been undertaking the accumulation of supplemental data sets from prioritized references. We understand which references are likely to contain meaningful supplemental information, including and especially large data sets of variant information. We have so far amassed a data set of about 10,000 supplemental materials. We have not yet made those available in the software. It is a data set that we can draw on for the purposes of panel design, however, and it would be something that we would draw out in the initial client conversation. We do not have geo data, which if I'm not mistaken, is the gene expression omnibus expression level data for these biomarkers. We don't yet have that data in any of our automated processes, but if that's a predicate for engaging with us, it's certainly something that we can have a conversation around.
So before we left the subject of ACMG, I was thinking maybe you might want to give these guys a little heads up on something that we're working on that's pretty exciting.
You can tell Candace is in marketing. Thanks, Candace. So I didn't show it in the screenshots for the software. It's something that we have been working on for several months now in concert with some of our development partners that we're going to be launching coincidentally at the ACMG trade show coming up here in a matter of several days, actually. We have in the professional edition of Mastermind, the capability of prioritizing references along the lines of ACMG classification criteria. So those aspects that I talked about, functional studies, case reports, whether there's any evidence or information in the literature to support this variant having loss of function consequence for the gene and its association with a particular disease pathogenesis. So that's something that we're launching here in a matter of days at the ACMG conference. Again, a feature which will be available only in the professional edition of Mastermind.
Great. So in closing the questions portion, I just have to say pathogenicity.
You got it.
Yeah, thanks. I want to thank you all so much for joining us today. I will be sending a follow-up email with a link to the recording of this webinar and the white paper Mark discussed in the next week. Be sure to visit us at genominon.com and follow us on all social media at Genominon. If you're interested in using our database in the way Mark demonstrated here, please email us at hello@genominon.com and have a great day.